This is going to be a partial key to the test review for cell structure and function. It's number one, a section of cell membrane, just the phospholipids. So one phospholipid, we would have a head and two tails, and the cell membrane is a bilayer, so double layer of phospholipids with the tails on the inside and the heads on the outside. The hydrophobic region is the tails, and the hydrophilic region is the part with the heads. Remember, hydrophilic means loves water, and it's watery outside the cells. Your blood and body fluids are watery, so the heads will orient facing the watery outside environment, and the hydrophobic non-polar tails will orient together on the inside away from the watery areas. Let's see, a fluid mosaic model. Make sure you understand both the mosaic, so lots of small parts, and the fluid part, meaning they're able to sort of move and slide past each other. So different types of transport are important. Let's first look at active versus passive. So active transport uses ATP energy. Passive transport doesn't. It just kind of happens by itself. So anything that is diffusion is going to be a passive transport. So both simple and facilitated diffusion, as well as osmosis, does not require energy of the cell. Exocytosis, endocytosis, and use of a pump, those are all active transports. Types of molecules: for simple diffusion, which means straight through the membrane, must be small, must be nonpolar, and the main examples of those are oxygen and carbon dioxide. Facilitated diffusion, meaning using a sort of protein channel to be able to move through, still needs to be small, but it could be something that's polar or an ion, like glucose being polar, or various different ions having a charge. Exocytosis is used for large molecules, like proteins would be the main example of that one. Endocytosis, also used for large molecules, but exocytosis is molecules going out, and endo would be molecules going in. So again, that could be proteins, or it could be something as big as an entire bacteria, or maybe food if it is like a amoeba or a single-celled organism. Osmosis is only used for water. Osmosis is just the movement of water through a cell membrane. And the sodium-potassium pump would be, again, well, in sodium-potassium pump, it's sodium and potassium that are being moved, but these pumps are usually going to be pumping ions. Other kinds of pumps might be pumping water or other things like that. But the key to this is going from low concentration to high concentration. So it's making things more concentrated, which requires energy to force those molecules closer together. Doesn't happen naturally by diffusion. Diffusion is only from high concentration to low concentration. Right, tonicities, isotonic would be same concentration in and out. Water would move in and out evenly from the cell. Hypotonic solution would be higher concentration of dissolved stuff inside the cell, lower concentration outside, so like pure water in a cell. That will cause water to move into the cell. Hypertonic, higher concentration outside the cell, so like a really salty solution, that will cause water to move out of the cell. For all of these cell parts, review your notes about them. Smooth endoplasmic reticulum, for some reason, people seem to have trouble remembering that one. You're not going to need to draw these on the test, but you will need to be able to recognize them if you see a picture of them in a cell, so make sure that you can do that. Plant cell wall is located outside of the cell membrane. Endosymbiont hypothesis was that idea that mitochondria and chloroplasts in particular probably were originally their own single-celled organisms, which then got sort of absorbed by another cell and became part of that cell and started working for that cell. Evidence for that includes all the ways that mitochondria and chloroplasts are similar to single-celled prokaryotes like bacteria. So things like they have their own DNA, their DNA is similar in structure to bacterial DNA, they have their own membrane, their own compartmentalization, they reproduce by dividing, they have some similar proteins and ribosomes to bacteria. All of those support the idea that they at some point were a cell like a bacteria. Difference between prokaryote and eukaryote. If you remember that eukaryote is you, right? So you are made of eukaryotic cells, as are all animals, plants, fungi, all multi-celled organisms. And then the prokaryotes are mostly bacteria. So the first type of cell to develop would be, this is kind of typoed weird. So this should be here, this should be here, this should be here. Sorry if that was confusing. I didn't realize these got slid over until I saw this. But first type of cell to develop is the prokaryote. They're the most simple. Contains membrane-bound organelles is the eukaryote. So all of those organelles other than ribosomes and a cell membrane, pretty much all of those are only in eukaryotes. Contains ribosomes and genetic material would be both. Genetic material stored in a nucleus, that is the main key component of eukaryotic cells, that they do have a nucleus surrounding their DNA. Animal cells are eukaryotes. Fungal cells are also eukaryotes. Bacterial cells, prokaryotes. Uses compartmentalization, eukaryotes. So only eukaryotes have internal membranes breaking the cell up into different compartments. Has a cell membrane is both. All cells have to have a membrane. Has one or more linear chromosomes is eukaryotes. So eukaryotes' chromosomes are in lines. Prokaryotes' chromosomes are in circles. Purpose of compartmentalization, again, is breaking up the cell into different compartments, different parts, so that chemical reactions can happen in their own, separate area, so that you don't have things interacting that you don't want interacting. You can store certain things in certain parts. It really increases the efficiency of the cell and what the cell is able to do. It also increases surface area inside the cell. Water potential, so our negative ICRT equation, had a lot of practice with these. Water potential of 0.18 molar sodium chloride solution. So sodium chloride salt has a ionization constant of 2, so negative 2 times 0.18 times 0.0831, which is always R, room temperature 298 degrees Kelvin. So we get negative 8.9 bars. If I place a piece of potato in the solution and find that the potato loses mass, what does this tell me about the water potential of the potato cells? So if the potato lost mass, that means that water went out of the potato, which means that the water potential of the potato cells must have been higher than negative 8.9 bars because water will move from high water potential to low water potential. So I would guess that the solution is most likely hypertonic to the potato cells because water went out. Cell communication, reception, transduction, response. Most of what you need to review and practice for cell communication is the actual practice AP questions, so the ones that we did in class that were multiple choice, as well as the ones on the AP classroom website are going to be the best place to review and practice sort of analyzing those examples and going through the process. The purpose of second messengers is to amplify and spread the signal. So those are when there's like a whole bunch of like a little molecule or maybe an ion released into the cell. For example, potassium ions could be a second messenger. There was another one called CAMP that was in there. Protein kinase is an enzyme that phosphorylates another protein. And phosphorylation is taking the phosphate from ATP, so it's essentially energizing or activating that protein. And the phosphorylation cascade is when you have one phosphorylating the next one, which phosphorylates the next one, and then the next one, and the next one, sort of on down the line. You can look up the structure of those receptors. And quorum sensing is, again, how bacteria count each other. So essentially, the bacteria send out these little chemical molecules, and when there's a lot of bacteria, there's a high concentration of these little chemical molecules which bond to receptors on the bacteria, and if the bacteria sense a high enough concentration of the molecules, they sort of know there's a lot of other bacteria around, and then they can do a coordinated response, do something all together like glow like the bacteria in that squid, or attack like a pathogenic bacteria. Positive and negative feedback. So positive feedback, think the snowball rolling down the hill, bigger and bigger response. Negative feedback, something's out of homeostasis, a response happens in order to correct that imbalance, and then the response is turned off once the imbalance is corrected. So example of positive feedback was that one with the woman giving birth, where it just gets stronger and stronger contractions until the baby is born. Much more common is negative feedback. For example, you feel cold, you start shivering as a response to feeling cold, and once you get warmer again, the shivering stops. Right, so that's going to be the end of this video. If you have any questions left over, please do bring them to me before the test.